Hello friends, welcome back to our channel NEET Biology Expert. I am Dr. Parveen, your biology professor. In this lecture series, we are studying about asexual method of reproduction. That is the chapter 1, reproduction in organisms from the plus 2 syllabus. Okay. In our previous lectures, we have covered two important types of asexual reproduction. One was fission and the another one was sporulation. These two topics we have covered in our previous lecture, and in this lecture we are going to study about the other major methods of asexual reproduction, that is budding, fragmentation, apolysis, gemmule formation and regeneration. Alright, let's get started. The first one is budding. Budding is a major method of asexual reproduction in unicellular organisms. For example, you see yeast. The biological name for yeast is Saccharomyces cerevisiae. Okay. And also in Hydra. What is Hydra? Hydra is a freshwater living organisms which we could see in the temperate and tropical conditions. So, Hydra, yeast and also Leuco Selenia. This is a species and that is common name is sponges. Sponges we could see in the marine water. You could have seen this in the Discovery Channel and all. No, underground marine water. In the deep oceans we could see this organism sponges. So these three organisms are a very good example for budding method of asexual reproduction. Let us see one by one. So what is yeast? The biological name for yeast is Saccharomyces cerevisiae. It is commonly called as baker's yeast. What is baker's yeast? This yeast, for example, you see in this picture a spoonful of dried yeast. Every individual molecule is a yeast. Okay. This is commonly used in bakeries, in homes to make bread, bun, naan and all. So what you have to do, just soak this yeast in a lukewarm water with some salt or some sugar and mix this with your flour, and after some period of time what will happen, the yeast ferment the carbohydrates or the starch in the flour and convert this into carbon dioxide. That's why the size of the flour gets bulged. Okay. So yeast is commonly used in the bakeries. So that's why it's called as dry yeast or baker's yeast. This yeast is a unicellular organism which do reproduce by budding method of reproduction. Look at this picture. This is the yeast cell under microscope. So this is a parent yeast cell. So when this yeast wants to reproduce, what happens? The parent cell suddenly bulges in one corner and produces the bud. The first stage is like this. So this is the nucleus present inside the cell, and what happens? It bulges at one direction in one corners. That means the daughter cells is about to develop. So that small daughter cell, the initiating cells are called buds. Okay. Then what happened? The bud slowly grows. So like this, when it was initially it was a small one, then it starts to grow gradually and finally it gets released into a individual daughter cell. Okay, now this cell will again make this reproductive cycle again and again. Okay, so this is a common method of reproduction in the yeast cell. So let us see with some diagrammatic explanation. So this is an adult yeast cell. So when it wants to divide, they divide by mitotic cell division. We know mitosis and meiosis, right? This organism, when they want to divide or produce asexual method of reproduction, they do by it mitotically. See, any organism when they are doing this binary fission or budding, what happens initially? Initially, the nucleus gets separated. That is called karyokinesis, separation of the nucleus. The second stage is cytokinesis, separation of the cytoplasm. Same thing happens here also. So, initially, the mother cell produces a bud, that is a small daughter at one end, and it slowly divides mitotically, whereby it shares its nucleus with the daughter cell. See, these green color things are called centrioles, okay, which are important for the mitotic cell division. And finally, the daughter cell gets separated or detached from the mother cell and it makes an individual life, okay. So this is the method of reproduction in the yeast, which we call as budding. So in this thing, a peculiar thing happens in the yeast. Not only one daughter cell will produce, sometimes it produces a chain of daughter cells. That's we called as chain of buds, okay. See, this is a mother cell, from the mother cell and another daughter like this. So one mother cell here produces 3 daughter cells simultaneously. This is called chain of buds. Right. So let us see what is the definition for budding. Budding means the parent body produces one or more buds. That's what I told. No, not only one bud. Sometimes it produces one daughter or sometimes it will be more than one. So it produces one or more buds and each bud grows into an individual young ones. Then the buds separate from the parent and leads to a normal life. So this is called budding, okay. Here this budding is due to the cell division which occurs at one particular site. See, for example, if this is a mother cell, here this is the nucleus. The cell division, for example, if it occurs here in this direction, from this area the daughters will be produced, the buds will be produced. So that's what we call the cell division at one particular site. This will not happen randomly. For example, if this is a yeast cell, one will not produce bud here and another bud will not be produced here, another bud will not be produced here, not like this. So once it starts the cell division will occur at one particular site. So this is very important in the yeast cell. The small bulb-like projection which is coming out from the yeast is called bud. We know this, right? That small daughter cell which comes out from the mother is called bud, right? The next important example for budding method of reproduction is Hydra. This is Hydra. What is Hydra? Hydra is a freshwater organism which we could see in the tropical and temperate countries. This is a very important organism which is of very much interested for the scientists to study. I will explain you why. Okay. So now this Hydra, it adopts 2 methods of asexual reproduction. One is by budding and another is by regeneration, which we will going to study later. So first method in budding, what Hydra do? First one is, see, look at this picture. This is an adult Hydra. This is the base of the organism and this is the mouth part, and these small structures, they are called tentacles. What is tentacles? They are the sense organs for the organisms. For example, you compare this with a cockroach. Cockroach has 2 long tentacles, right? They are the sense organs. When the cockroach wants to move in one direction, it first stretches its tentacles and notices if there is any distraction or whatever is present in that direction, and from that it starts moving or move from one place to another. Okay, so like this Hydra sense organs are these hair-like structure called tentacles. Okay, so when this Hydra wants to reproduce asexually by the means of budding, what happens? Same like in yeast, and bud is formed in one end. Okay, so see here, this Hydra is having 2 layers of the cell. One, the outside layer is called ectoderm and the inside layer of the cell is called endoderm. So what happens first, this ectodermal layer, the outside layer of cells of Hydra, it gets bulged and they forms into a small bud. Okay, next step, the endodermal layer, the inside layer, it pushes out and it forms a big bud. Okay, then this bud enlarge and it develops a mouth and the tentacles, and finally the young ones get detached from the parents from the base itself and it leads an independent life. Okay, so this is a very good example for budding method of reproduction, that is Hydra. So look at this picture. This is an adult hydra before reproduction. So here I told you, this ectoderm starts bulging and finally it forms a bud in this direction. So it will develop and gets detached. So this is the hydra budding method of reproduction. So hydra and yeast both does this budding method of reproduction during favorable environment, when the conditions of these organisms are satisfied. If they have enough food, good temperature, good environment, in those conditions they adopt this method of reproduction. You should always remember one thing: for every organism which reproduces by asexual, there is variation between every organisms. Okay, it starts from binary fission till regeneration. Whatever we are going to study, you have to write down and study completely, like which method of reproduction which organisms are adopting and during which condition they are adopting that. For example, amoeba reproduces by binary fission during favorable environment. Same amoeba reproduces by sporulation or encyst formation during unfavorable environment also. Okay, so these minute points you should remember for every examples, right? So that's why similarly hydra and yeast, they reproduce by budding during favorable environment, right? Another example for budding method of reproduction is sponges. So one peculiar species is Leuco Selenia. This is a species of a sponge marine organism where you see this is an individual parent cell. This parent cell starts outgrowth and begins to grow at the base here, and it forms a bud. Small bud starts to form and the buds continues to grow. Okay, finally the bud breaks off and attaches to the new surface. So when this organism reproduces and gives buds, the bud separates from its bases. Then only it will go to another place and it needs a firm environment to sit, and it will start its own life cycle. So that's what it detaches completely along with the base, breaking off from the base. Okay. Here this osculum means the mouth of the sponge. For example, you see this picture, we usually see the sponges in pictures like this. No, this is a group of sponge actually with more buds. The parent cell is fully surrounded by daughter buds. Okay, this is the picture. Initially the parent cell will be like this, individual or a single stalk like that, and when it reproduces by budding, it will produces a group of daughter cells. So this is budding method of asexual reproduction. Okay, so these hydra, yeast and the sponge, these are examples for exogenous budding. What is exogenous? Exo means outside. There the buds are produced outside the parent cell. So that's why they are called exogenous budding. There is another method of budding that is called endogenous budding. Example for this is Noctiluca. Noctiluca is a dinoflagellates which shows bioluminescence. This we could see in some seashores. Okay, so this is a organism under microscope. This is the diagrammatic picture of the organism. So what happens, this Noctiluca dinoflagellate also reproduces by means of budding, but the buds are present inside the parent cell. Okay, Noctiluca buds are formed inside the cytoplasm. So here in the cytoplasm, there will be more buds in the organisms. They remain within the body of the parent cell and they will get released whenever the environment is favorable for them. Okay. So this is called endogenous budding in this organism. Understand? Budding. The next method of reproduction is called fragmentation. So what do you mean by fragmentation? Fragmentation means the body breaks down into distinct pieces or fragments. This occurs in some multicellular organisms, not in unicellular organisms. In multicellular organisms, what will happen? The body of the parent cell gets into pieces, gets into fragments. and each fragment then grows into an adult. The young ones grows into an adult. Okay, so this method of asexual reproduction is called fragmentation. Example for them are two important example, sea anemone and Spirogyra. Sea anemone, again you could see this in the marine environment, marine water, and Spirogyra is a freshwater algae. So these two are the example for fragmentation. Let us see the first one, sea anemones. We might have seen all this in the Discovery Channel, right? They are beautiful underwater organism. Can you say whether this is a plant or an animal? It looks like plant and it also looks like animal. Genetically it looks like plant and also looks like animal. Half plant and half animal. Okay, so very peculiar organism that is called sea anemone. See, very beautiful bright colors they are, white, pink, red, purple, many colors we could see in sea anemones. Okay, so sea anemones, when it wants to reproduce asexually, what happens? This is an adult organism. This is an adult or a parent sea anemone. When it wants to reproduce, it breaks a part of its body. It separates one part of the body by itself. It means it gets its body fragmented, and from that separated part the young one starts to develop, right? So the sea anemone, it separates its part. No, that part is called pedal lacerate. The separated part of the sea anemone is called pedal lacerate. So from the pedal lacerate, the young anemone gets developed, right? So this is one method of example for fragmentation. The second example is very famous example is Spirogyra. What is Spirogyra? Spirogyra is a freshwater algae. See this? These are the algae. So much Spirogyra in one particular area. This is a freshwater algae. This algae we could use for medical purposes. From this algae we get antibiotics, antiviral components like this. It is more than 400 species are there in this Spirogyra. Okay, so this is having chlorophyll. You know what is algae, right? They are small, almost plant-like structure. They have chlorophyll and they produces their own starch with the help of photosynthesis. So see this strands, individual strands of this Spirogyra. This like this, there is one structure is there. No, these are the strands of chlorophylls in this organism. Okay, this is very zoomed microscopic picture. So this organism, they reproduces by means of fragmentation. How? For example, if this is one strand of Spirogyra, when it wants to reproduce, it gets fragment. It cuts itself into several pieces at particular intervals, and that each fragment gets developed into an adult organism. So this is a peculiar example for fragmentation method of reproduction. Okay. The next method of asexual reproduction is called apolysis. The apolysis, very good example is pig tapeworm, or we can call this pork tapeworm. So what is the biological name for this organism? This is called TNSCERT. TNSCERT. There is another tapeworm that is called Taenia sanguinata. They are called as beef tapeworm, and this is pork or pig tapeworm. See this picture. This is the head of this worm that is called scolex. This is the neck, and all these long structures are their tail or the body parts. Okay, so let us see how it reproduces. So this is scolex, the small part at one corner, that is the head. Head is called scolex, neck, and these structures. These body structures are called proglottids. See, this proglottids has two stages. Whichever area which is closer to the neck, they are called immature proglottids. Whichever areas very farther, that is at the posterior end, very much end of the organism, they are called gravid proglottids. In between them are called the mature proglottids. Okay, so three stages of proglottids is present in this organism. So what happened here? The gravid or the ripe proglottids are the oldest at the posterior end. I told you, no, these are called the gravid proglottids, or the oldest one, which are present in the posterior end, very extreme end of this organism. And the gravid proglottids are regularly cut off, either singly or in groups. See here, the organism either cut off these proglottids individually or in groups, like 2, 3, 4, like this. Okay, it releases of that particular area from its body. So this process is called apolysis. So why apolysis occurs in the taenia? It is a very important method of its reproduction asexually. How? Because you see here, this is the life cycle of this taenia solium. It has two stages or two hosts. The first host is man and the second host is the animal, particularly pig. Okay, so this is the taenia, the tapeworm. It gets entered into the human being through contaminated food, particularly uncooked pork meat. Okay, so they sit and multiply in the small intestine. In the small intestine, when this organism develops into a mature one, it releases its proglottids. The large proglottids are shed. Okay, so that comes out in the feces of the human. So when it gets mixed with the environment, for example, in the lawn, in the grass, in the outside areas, when any pig or any animal feeds on the grass is contaminated with a human fecal material containing this proglottids, the organism enters into the animal body, it gets multiplied. Okay, again what happens? We will eat that pork, the meat. If it is undercooked or uncooked, again the cycle repeats. So this apolysis is helpful in transferring the developed embryos from the primary host, that is from the man, to find the secondary host, that is the pig. So the organism wants to take two life cycle. Okay, so from man to the pig. So how it is going? Through this proglottids method. So this method of reproduction is called apolysis, which occurs in taenia. The next one is gemmule formation. Gemmule formation is very good example for this is sponge, freshwater sponge. So one species is called Spongilla lacustris. See, this is a very happy vegetative sponge. This you could see on the stagnant fresh water. See here, these all green bunch of organisms. These are called as a sponge in the fresh water. Okay, so what happens when this organism are undergoing an unfavorable environment? Suppose in that freshwater pond, the water is getting dried, there is no nutrients, there is no good environment of temperature like this. For this sponge, it adapts to another method of reproduction, that is called gemmule formation. Okay, this occurs during unfavorable environment. That is a peculiar mode of asexual reproduction. Okay, so these structures are called gemmules. Internal buds, very important, the word internal buds. So if you zoom this, it will look like this. See, these small round structures are called internal buds or gemmule. So let us take one bud, one small bud, and put a cross section. What we could see? We could see structure like this. So what is gemmule? Gemmule is a hard ball. These every one thing is called a gemmule, and it is a hard ball. What it contains? It contains an internal mass of food-laden arachiocytes. So when you make a cross section of the gemmule and look at inside, you could see very minute particles. These are the food storage. Okay, so these organisms, they want to withstand this extreme condition, or when there is no food, when there is a drought like this condition, they want to live. After that, they will not get died immediately, because they store their excess food in structure called arachiocytes. Okay, this is a resting stage, right? So what will happen when the conditions become normal? Look here, during unfavorable environment, they produces these gemmules. Again, when the environment turns favorable, when it rains, the pond is full. So like this, when the environment is favorable, it changes its structure into a vegetative cell. So like this, these two are interchangeable morphological structures during favorable and unfavorable environment. So this method of asexual reproduction in sponges is called gemmule formation. So let us see the last part of asexual reproduction in organisms is the regeneration. So very good example for regeneration method of asexual reproduction are Hydra. Again you see Hydra. Hydra reproduces by budding. Initially we saw no budding, and also it undergoes regeneration. Another example is a starfish and lizard, house lizard particularly. Okay. What is regeneration? Regeneration means regrowth. Re means repeat, growth means again growing. Okay. So regeneration means regrowth in the injured region. Where the organism is growing, whichever part the organism was injured, that injured part gets regenerated, gets regrowth. Okay. So that is the meaning for the word regeneration. So this was first studied in Hydra by Abraham Tremblay in 1740. So he only first noticed this regeneration is a method that is occurring in Hydra. Do you remember? Initially I told you Hydra was very much interested organism for many scientists. Why? Because they adopt this method of regeneration. Okay. Let us see what is that. This regeneration is classified majorly into two types. One is called morpholexis and another is called epimorphosis. Small difference between these two. Okay, basically these are regeneration only. First one, what is morpholexis? Here the whole body grows from a small fragment. So for example, if that organism has a cut, from the cut part the whole organism gets developed. Okay, very peculiar type of reproduction, or that is called regeneration. Very good example for this is Hydra and Planaria. Planaria is a flat worm, belongs to Platyhelminthes group. Okay, so they also have this regeneration. Even amoeba also has this regeneration capacity. Let us see example with Hydra. What is happening in Hydra during regeneration, that is morpholexis? When Hydra is accidentally cut into several pieces, each piece can regenerate. The lost parts will develop into a whole individual organism. Not only developing, they retain their polarity and their oral ends. So what do you mean by that? Let us see this example. Look at this. This is a Hydra. Okay, this black one is a Hydra. We are going to cut this Hydra in the middle. We are making to cut. The center part gets detached, right? So what will happen? This part which is having only the head will regenerate its tail or the base. See, from the head the tail parts grows and it becomes a fully regenerated Hydra. Okay. Similarly, the tail part which lost its head, it starts to regenerate and it will develop the head part. So, completely from the tail, a new Hydra develops, right? Not only that, look at here. The center part which lost its head and tail, both the head and the tail gets regenerated. So, like this, the Hydra is having this peculiar characteristics of regeneration. Okay. So because of this, scientists used to say Hydra is having no death. There is no mortality, no death for Hydra. Why? If you cut the Hydra into 100 parts, every part gets developed into a new Hydra. There is no natural death in Hydra. Okay, look here. Even if you cut this in a longitudinal section like this, both the left and the right direction, right side, they get regenerated into its original structure. Okay, so this is a very, very important and interesting character. That's why scientists wants to study Hydra more, how this organism is having this capability, what gene is responsible for this, or what mechanism is behind this. Can we adopt this for human being also? So like this, many researches are going on in Hydra. So here the whole body grows from a small fragment. That is very peculiar. The same thing happens in planaria worm also. So wherever you cut this organism, it will regenerate the whole body parts. Understand? So this is morpholexis. The whole body grows from a small fragment. The next method of regeneration is called epimorphosis. Here, replacement of only a particular part of the body parts, that is called epimorphosis. Not the whole body, a particular part only will develop. Okay, so that is called epimorphosis. The example for this are starfish and lizard. Okay, so what happened? Severed body parts can develop. That is called epimorphosis. Look at this starfish. If one arms of the starfish gets cut or detached accidentally, what will happen? After some period of time, the last part will develop. Okay, so this method is called epimorphosis in the starfish. In some starfish, not only that, the last part will have the ability to produce the whole organism also. This also occurs in the starfish. But the another example for epimorphosis is lizard, common house lizard. We call this as leopard gecko. Okay, house lizard. We might have seen this. Whenever we try to hit a lizard, it will lose its tail and run off from that place. Have you seen it? What will happen after some period of time? The tail will start to grow, particularly within 4 weeks to 60 days. In 2 months, the lizard, whichever lost its tail, this tail parts gets regenerated like this, you see. So this method of regeneration in lizard is called epimorphosis. Why this occurs in lizards like this? See, it involves dropping a section of the tail and gets regenerated. It is a part of defense mechanism. That is very important. So why this lizard, even if it loses its tails, it's living and the tails gets regenerated after some period of time, means this is a method of defense for this organism. So this is also called as self-amputation or autotomy. This is important. You should remember self-amputation or autotomy. What is self-amputation? For example, have you heard about Clostridium tetani or many other anaerobic bacteria which causes severe infections in human body? Okay, even in diabetes patients, when the diabetes is very severe, what will happen? If they have any wound in their legs, the wound will develop very much severely. Okay, so at that extreme condition, what doctors will do? They will amputate, surgically remove the legs of the patient, right? So why they are doing like this? In order to stop or prevent the infection getting spread into the major organs. Okay, that is called amputation. Amputation means surgical removal of a particular part, particularly limbs only. We cannot amputate internal organs, because amputation means even if we remove the organs, we could able to survive. That is called amputation. Okay, so generally legs and hands, these are the parts we usually amputate. Okay, so that method is called amputation. Here what happens? This lizard is self-amputating. It itself cuts its extremities. Okay, that is called self-amputation or autotomy. The casting of a part of the body, that is the tail of the lizard, under when the animal is during threat. Okay, when the animal is in the danger, they does this method. So this is an example for regeneration. Understand? So let us have a quick recap of this lecture. Reproduction, we are studying about asexual method of reproduction. Last two lectures we studied about fission and sporulation. Fission, there are 4 types: binary fission, multiple fission, sporulation, strobulation. Again, binary fission is classified into 4 methods based on which angle the fission occurs, as simple, irregular, transverse, longitudinal and oblique. Right? Sporulation, 2 example: chlamydomonas, which produces zoospores, and penicillium, which produces conidiospores. So in this lecture we have seen about budding. Budding, 2 types: exogenous budding and endogenous budding. Exogenous budding, present, the buds are present outside the parent cell. That is called yeast, hydra and sponges. Example: endogenous budding, example, Noctiluca. 4th method of reproduction is by fragmentation, whereby the parent cell it gets fragmented and each fragments develop into an adult cell. So example for this are Animal and Spirogyra. The next method is by apolysis, whereby the older proglottids are shed slowly and they will go and adopt a new host. So example for this is TNSCERT, pig tapeworm, right? Gemmule formation. Gemmule means inside spores or endospores or internal spores. Okay, so when the environment is unfavorable, the organism produces internal spores. They are called gemmules. Example for these are sponges. And last one is regeneration. Regeneration means the organism regenerates or redevelops a lost part. Either the full body gets regenerated or a particular part gets regenerated. Example: Hydra, starfish and lizard. Clear? Alright. So our next lecture will be about asexual method of reproduction in plants, that is vegetative reproduction in plants. Okay. If you like this lecture, please give a like and don't forget to subscribe to our channel and don't forget to press the bell icon. So whenever we post new lectures, it will reach you immediately. And please share this with your friends. See you in another lecture. Until then, take care. Bye.